What every parent should know: an independent investigation into vaccine ingredients. Disclaimer: this investigation presents factual information about vaccine ingredients and manufacturing practices for educational purposes. We are not providing medical advice or telling anyone whether they should or should not vaccinate their children. The decision about your child's healthcare belongs entirely to you. Our goal is simply to provide information that may not be readily available elsewhere so you can make the most informed decision possible for your family. At 38 years old, with a child on the way, I found myself facing a question I'd never seriously considered: should I vaccinate my baby? I received all my childhood vaccines without question. But times have changed, and the debate around vaccine safety has intensified. When I tried to research the topic, I encountered a problem. Neither official sources nor anti-vaccine sources provided verifiable, objective information I could trust. Both sides seemed to present selective facts that supported their predetermined conclusions. So I decided to conduct my own investigation. Instead of relying on interpretations and opinions, I went directly to the primary sources: peer-reviewed scientific literature, official FDA and CDC documents, and vaccine package inserts. All sources are linked throughout this article for your own review and verification. What I discovered in this months-long investigation surprised me and raised questions I believe every parent deserves to consider. This article presents my findings, not to tell you what to do, but to share information that might help you make a more informed decision for your own family. Starting with the basics: what's actually in vaccines? I began by examining the actual ingredients listed in official FDA documents and vaccine package inserts. Here's what babies receive according to the CDC's recommended schedule. DTaP vaccines, multiple brands. Daptasol, per half a milliliter dose, 1.5 mg aluminum phosphate, 0.33 mg aluminum less than or equal to 5 micrograms residual formaldehyde less than 50 nanograms residual glutaraldehyde 3.3 mg, 0.6% VV, 2-phenoxyethanol Infinrix, per half a milliliter dose, aluminum hydroxide, up to 0.625 mg aluminum less than or equal to 100 micrograms residual formaldehyde less than or equal to 100 micrograms polysorbate 80, tween 84.5 mg sodium chloride Vaxilis, DTaP IPV HIB-HEP, per half a milliliter dose, 319 micrograms aluminum from aluminum salts less than or equal to 14 micrograms formaldehyde less than or equal to 50 nanograms glutaraldehyde less than or equal to 50 nanograms bovine serum albumin multiple antibiotics and animal proteins. A critical scientific inconsistency discovered. While examining these ingredients, I discovered a puzzling inconsistency that raises questions about manufacturing transparency. This vaccine is marketed as containing only inactivated components, DTaP, inactivated bacterial toxins and purified proteins IPV, inactivated poliovirus HIB, conjugated polysaccharide, bacterial sugar HEP, recombinant proteins made in yeast, yet Vaxilis contains bovine serum albumin, yeast proteins, and multiple antibiotics. Industry explanations note that these ingredients are used for growing bacterial and recombinant proteins during manufacturing, with trace amounts remaining after purification. However, this raises important questions about why manufacturing processes require these contaminants and whether cleaner production methods are possible. Scientific fact: aluminum is not used in live, weakened viral vaccines, like measles, mumps, rubella, varicella and rotavirus because aluminum would kill the live viruses needed for immunity. Scientific fact: formaldehyde cannot be used in any of the live vaccines such as the MMR, rotavirus, varicella, chickenpox, and some flu vaccines because of its antimicrobial properties. It would kill the live viruses. Scientific fact: bovine serum albumin, yeast proteins, and antibiotics are specifically used when growing live viruses in cell cultures to feed the viruses and prevent contamination during live virus production. The question: if Vaxilis contains no live viruses and everything is pre-inactivated, why does manufacturing require ingredients typically associated with live virus production? Greater transparency about these manufacturing processes would benefit parents seeking clarity about what's being injected into their babies. By age 18 months, following the CDC schedule, babies receive a cumulative total from all vaccines combined, 4 to 5 plus mg of aluminum injected directly into muscle tissue, 200 to 400 plus mg of formaldehyde, multiple doses of polysorbate 80, 2-phenoxyethanol, and various antibiotics. These aren't theoretical amounts, these are the actual total quantities injected into developing babies when you add up all the vaccines they receive over their first 18 months of life. Aluminum, a neurotoxin in every shot. The first ingredient that caught my attention was aluminum. The scientific literature is clear: aluminum is neurotoxic. This isn't disputed. What surprised me was learning how aluminum in vaccines behaves differently from aluminum we might ingest. The science on aluminum toxicity. When we eat aluminum, from food or antacids, only about 1% is absorbed into the bloodstream, and the kidneys quickly eliminate most of it. The blood-brain barrier provides additional protection. But injected aluminum bypasses these natural defenses entirely. 100% enters the bloodstream. Recent research shows that aluminum adjuvant nanoparticles are carried throughout the body by immune cells called macrophages. These poorly biodegradable aluminum-coated particles injected into muscle are promptly phagocytosed in muscle and the draining lymph nodes, and can disseminate within phagocytic cells throughout the body and slowly accumulate in brain. Studies also show aluminum damages the blood-brain barrier, BBB, exerting pro-oxidant effects, activating microglia and neuroinflammation, depressing cerebral glucose metabolism and mitochondrial functions. The dose problem: to put this in perspective, a 6-month-old baby, 7 kilograms, receiving 3 milligrams of injected aluminum equals 0.43 milligrams/kg directly into muscle tissue. Due to 100% absorption versus 1% from food, this is equivalent to a baby consuming 300 to 400 milligrams orally. For comparison, this is 17 times more aluminum than safety guidelines allow when adjusted for body weight. Critical gap. While mainstream toxicology references consider these levels safe when examined in isolation, the effects of cumulative multidose exposure, particularly in the context of immature immune and detoxification systems, have not been studied in sufficient depth. This represents a fundamental gap in safety assessment. The safety study problem here is where things get concerning. Most aluminum safety studies in vaccines use aluminum containing placebos rather than truly inert substances. As one comprehensive review states, to really evaluate the safety of aluminum salts in vaccines, one would have to account for all known/potential immunological mechanisms involved with aluminum adjuvants. What are the potential negative outcomes due to that mechanism? What tests are needed to check for those outcomes? This comprehensive safety testing has never been done. Given that aluminum is an established neurotoxin that accumulates in brain tissue, why aren't there long-term studies examining cumulative aluminum exposure in developing babies? Why do safety studies use aluminum containing placebos, making it impossible to assess aluminum's true safety profile? Formaldehyde, industrial carcinogen in baby shots. The next ingredient I investigated was formaldehyde, a known human carcinogen that causes DNA damage, inhibits DNA repair mechanisms, and is linked to cancer. The DNA damage evidence scientific research shows formaldehyde causes DNA protein crosslinks, creates single-strand breaks in DNA, initiates DNA double-strand breaks, inhibits DNA repair mechanisms. Effects occur at concentrations as low as 25 micromolar. The misleading endogenous formaldehyde argument. Health authorities often claim that babies naturally have more formaldehyde in their bodies than vaccines contain, so vaccine formaldehyde is safe. This argument is scientifically misleading. The body produces formaldehyde as part of normal 1-carbon metabolism, but it's immediately processed by specialized enzymes, ADH5, and converted to useful formate. This is a controlled process where formaldehyde exists only as a brief intermediate before being detoxified. Research shows formaldehyde detoxification by ADH5 supplies 1-carbon units for nucleotide synthesis and is essential for DNA synthesis, protein methylation, and cellular energy production. Injecting formaldehyde directly bypasses this controlled metabolic pathway, potentially overwhelming local detoxification capacity. It's the difference between a chemical factory producing exactly what it needs through refined processes versus flooding it with raw toxic material. The synergistic toxicity concerns. Studies on anthrax vaccines showed that aluminum plus formaldehyde combinations created significantly higher immune responses than either substance alone, suggesting synergistic effects. While researchers celebrate enhanced immune response, they haven't studied the enhanced toxicity, DNA damage, or long-term neurological effects of this combination. Considering that formaldehyde is a proven carcinogen that damages DNA repair systems, and given that no long-term studies track cancer or neurological outcomes in babies injected with formaldehyde, shouldn't we be more cautious about exposing developing infants to this industrial toxin? Other concerning ingredients: polysorbate 80. Blood-brain barrier risks. Polysorbate 80 is used as an emulsifier in many vaccines. Research in animal studies shows it can increase blood-brain barrier permeability, potentially allowing other ingredients direct access to the developing brain. While these studies used higher doses than found in vaccines, the question remains, why give multiple doses of a substance that can compromise brain barriers to babies whose blood-brain barriers are still developing? Recent studies also link synthetic emulsifiers like polysorbate 80 to gut inflammation, food allergies, and increased cancer risk. 2-phenoxyethanol, nervous system toxin. This preservative was banned by the FDA for use in nipple creams because it can cause central nervous system depression. Yet it's injected directly into babies. Manufacturing residues, evidence of non-sterile production. The presence of antibiotics like neomycin and animal proteins like bovine serum albumin reveals that vaccines are manufactured under non-sterile conditions. According to the FDA, antibiotics are used in some production methods for making inactivated influenza virus vaccines. They are used to reduce bacterial growth in eggs during processing steps, because eggs are not sterile products. This means vaccines are produced in contaminated environments that require contamination-preventing ingredients. If vaccines were made in truly sterile facilities, these antibiotics wouldn't be necessary. The vulnerable timing, attacking immature systems. The current vaccine schedule targets babies when their immune and detoxification systems are least developed. Immune system immaturity research shows that newborns have diminished immune responses to the types of antigens in vaccines. The immune system doesn't fully mature for months to years after birth. Most protective maternal antibodies transfer during the third trimester, so premature babies are especially vulnerable. The premature baby scandal. The most vulnerable babies receive the same toxic doses as full-term babies, same aluminum dose whether baby weighs 6 pounds or 20 pounds. Studies show 47% of premature babies have cardiorespiratory events after vaccination. No adjustment for gestational age, birth weight, or immune system maturity. Why do we use a one-size-fits-all approach when babies' weights, genetic backgrounds, and developmental stages vary dramatically? Why don't we test for genetic variations in detoxification capacity before injecting neurotoxins? The legal immunity problem. Why safety isn't the priority. In 1986, Congress passed the National Childhood Vaccine Injury Act, which removed vaccine manufacturers from civil liability for vaccine injuries, creating a legal immunity that explains much about current vaccine safety problems. How legal immunity works. No vaccine manufacturer can be sued in civil court for damages from vaccine injuries. Victims must go through a special federal vaccine court with caps on damages. The fund is paid for by a tax on vaccines, not by manufacturers. In 2011, the Supreme Court ruled that vaccine manufacturers are shielded even from lawsuits for defective vaccine design. The revolving door, regulatory capture. Research reveals systematic corruption in vaccine oversight. 54% of CDC employees who left the agency from 2004 to 2020 went to work for pharmaceutical companies. Internal FDA emails show the agency explicitly tells departing employees they can work behind the scenes to influence agency decisions. After joining pharmaceutical companies, all 8 new members of the CDC's Vaccine Advisory Committee have direct financial ties to vaccine manufacturers with no legal liability and regulatory agencies staffed by industry insiders. What incentive do manufacturers have to use the safest possible ingredients? Historical manufacturing disasters. Vaccine contamination isn't theoretical, it has happened repeatedly. SV40 contamination, 1955 to 1963, the most devastating vaccine contamination in history. An estimated 10 to 30% of polio vaccines were contaminated with simian virus 40, a cancer-causing monkey virus. 98 million Americans were potentially exposed to a virus that causes cancer in laboratory animals. SV40 DNA has been found in human brain tumors, bone cancers, and mesotheliomas. Some vaccines from Eastern European manufacturers contained infectious SV40 even after 1961. The FDA's own assessment concludes, the committee is not aware of a comprehensive system that is transparent and clearly understandable to, and accessible by, the public for preventing vaccine contamination. If contamination happened before, why should we assume current manufacturing is perfect? The presence of preservatives and antibiotics in current vaccines suggests ongoing contamination issues. The pattern of vaccines that caused more harm than good. During my investigation, I discovered several historical examples where vaccines caused more damage than the diseases they were meant to prevent. The 1976 swine flu vaccine emergency legislation for the National Swine Flu Immunization Program was signed into effect in mid-April 1976, bypassing normal safety protocols. More people died from the vaccine than from the swine flu. The vaccine was pulled from the market. Not only did the CDC approve this vaccine without even valid testing proving that the swine flu existed, they ran media campaigns claiming many famous people took the shot when they hadn't. The real victims of this pandemic were likely the 450-odd people who came down with Guillain-Barré syndrome, a rare neurological disorder, after getting the 1976 flu shot. On its website, the CDC notes that people who got the vaccination did have an increased risk of approximately 1 additional case of GBS for every 100,000 people who got the swine flu vaccine. Pandemics and the narcolepsy epidemic. The 2009 H1N1 influenza pandemic left a troubling legacy in Europe. More than 1,300 people who received a vaccine to prevent the flu developed narcolepsy, an incurable, debilitating condition that causes overpowering daytime sleepiness, sometimes accompanied by a sudden muscle weakness in response to strong emotions such as laughter or anger. The manufacturer, GlaxoSmithKline, GSK, has acknowledged the link after the pandemic's vaccination campaign in 2009 to 2010. The risk of narcolepsy was increased 5 to 14-fold in children and adolescents and 2 to 7-fold in adults. According to observational studies, the risk of narcolepsy was elevated for 2 years after the pandemic's vaccination. A 2018 meta-analysis found that during the first year after vaccination with pandemics, the relative risk of narcolepsy increased 5 to 14-fold in children and adolescents and 2 to 7-fold in adults, and the vaccine attributable risk in children and adolescents was approximately 1 per 18,400 doses of vaccine. The 1960s RSV vaccine disaster. In 1962, a formaldehyde inactivated RSV vaccine was tested in children for the first time by doctors at Children's Hospital in Washington, D.C. It flopped. Not only did the vaccine fail to protect children, but when vaccinated children later encountered wild RSV, they developed severe enhanced disease. At least 2 children died from this vaccine-enhanced illness. Safer alternatives, they exist but aren't used. Through my research, I discovered that safer alternatives exist for every toxic ingredient in vaccines, and many are already FDA approved for other uses. For aluminum, immune enhancement, calcium phosphate, already used in some vaccines with excellent safety profile. Squalene, a compound from the body's normal cholesterol synthesis pathway, already used safely in approved vaccines. Natural plant saponins, QS21 isolated from Calea saponaria trees, is already used as an adjuvant with proven safety for formaldehyde preservation. Trehalose, multiple studies show vaccines stabilized with trehalose maintain efficacy for months at high temperatures, with trehalose stabilized vaccines showing improved protective efficacy compared to unstabilized versions. Ascorbic acid, vitamin C. FDA states that ascorbic acid is generally recognized as safe for use as a chemical preservative in foods and as a nutrient for polysorbate 80, emulsification. Lecithin, research shows lecithin can be completely metabolized by humans and is non-toxic. Studies found lecithin promoted gut microbiota diversity and enhanced beneficial gut bacteria, opposite to polysorbate 80's effects. The 25-year timeline. How long have safer alternatives been available? Here's what shocked me most. These safer alternatives haven't just recently been discovered, they've been available and studied for decades. Trehalose as a safe stabilizer, FDA GRA approval. Trehalose was affirmed in the US as generally recognized as safe, GRAS, May 2000. In October 2000, the US FDA gave a letter of no objection to a GRA notice, GRN 000045. Earlier approvals. Trehalose was approved as a food ingredient in Korea and Taiwan in 1998 with no use limits. Trehalose was previously approved as a novel food in 1991 in the UK for use as a cryoprotectant for freeze-dried foods. Research on trehalose for vaccine stabilization. Early vaccine research. Studies from 2008 to 2009 showed trehalose stabilized vaccines had superior protective efficacy compared to unstabilized versions. Extensive research period. Multiple studies from 2019 to 2024 demonstrate trehalose can stabilize vaccines for months at high temperatures, with some vaccines showing better efficacy than current formulations. Current therapeutic use. In 2021, the FDA granted Fast Track designation for SLS005, trehalose injection, for treating spinocerebellar ataxia, showing trehalose is already approved for direct injection. What this means: trehalose has been FDA approved as safe for 25 years, since 2000. And extensive research over the past 15+ years has proven it works better than current vaccine stabilizers. Yet childhood vaccines still use formaldehyde. The selective application of regulatory barriers. During my investigation, I discovered a troubling pattern in how regulatory agencies apply safety standards. When examining why safer alternatives haven't been implemented despite decades of evidence, I consistently encountered claims about complex regulatory barriers and the need for extensive testing. Yet this same system has repeatedly fast-tracked products with minimal long-term data when it served other interests. The pattern of rushed approvals. The 1976 swine flu vaccine, emergency legislation for the National Swine Flu Immunization Program, being signed into effect in mid-April. By the time immunizations began on October 1, though, the proposed epidemic had failed to emerge. With President Ford's re-election campaign looming on the horizon, the campaign increasingly appeared politically motivated. COVID-19 vaccines. The COVID-19 vaccine from Pfizer-BioNTech, which was the first to receive an EUA, was under clinical development for 6 months before it submitted its EUA. An EUA was granted in less than a month. Full approval was issued 8 months later. Meanwhile, FDA approval guidelines recommended studies continue as long as feasible, ideally at least 1 to 2 years, but EUA required only a median follow-up duration of at least 2 months. The COVID risk context. When examining whether the COVID emergency justified bypassing normal safety protocols, the mortality data provides important context. The overall death rate for the seasonal flu in 2019 was around 0.1%, and the US COVID was more severe than seasonal flu, with recent 2023 to 2024 data showing patients hospitalized for COVID-19 had a 5.70% death rate versus 4.24% for seasonal influenza, about 35% higher mortality in hospitalized patients. However, researchers found that for the United States as a whole, 17 years of flu and pneumonia deaths are needed to match 3 years of COVID-19. This means COVID killed approximately 5 to 6 times more people annually than seasonal flu, a significant but not unprecedented difference when compared to other health emergencies we manage routinely without declaring emergencies. The contradiction exposed when introducing new vaccines with minimal testing, emergency use authorization processes in months. Public health emergency justifies bypassing safety requirements. Political pressure to move quickly. 2 months of follow-up data considered sufficient when improving existing vaccines with safer ingredients. Complex regulatory barriers prevent changes. Claims that safer alternatives don't exist, despite 25 years of evidence. Suddenly concerned about proper testing and long-term studies. Years of additional research allegedly needed. What this reveals. This selective application of regulatory standards suggests that safety and proper testing are invoked primarily when they serve to maintain the status quo. When economic or political incentives align with rapid approval, the same agencies that claim to be bound by rigid safety protocols demonstrate remarkable flexibility. The fundamental question: if regulatory agencies can authorize completely new vaccine technologies in months during an emergency, why can't they approve the use of safer, well-studied ingredients in existing vaccines over the course of 25 years? The economic logic, why this makes perfect sense. When I examined the economic incentives, everything became clear. 1. Legal immunity removes financial incentive to improve safety. 2. Regulatory capture ensures agencies serve industry interests. 3. Mandated market creates guaranteed profits regardless of product quality. 4. Toxic ingredients are cheaper than safer alternatives. This isn't healthcare, it's a protected business model where the most vulnerable customers, babies, can't choose alternatives, and manufacturers face no consequences for using the cheapest possible ingredients. Following the science, what the evidence shows. After months of investigating peer-reviewed research and official documents, the evidence points to several troubling conclusions. 1. Current vaccine ingredients include proven neurotoxins and carcinogens injected into babies with immature detoxification systems. 2. Safety studies are fundamentally flawed, using aluminum-containing placebos and short follow-up periods. 3. Safer alternatives exist for every toxic ingredient but aren't used. 4. Legal immunity and regulatory capture remove incentives for safety improvements. 5. The most vulnerable babies, premature, low birth weight, receive the same doses as full-term babies. 6. Long-term safety studies comparing vaccinated versus truly unvaccinated children don't exist. Questions that demand answers. As a parent facing this decision, I'm left with questions that seem reasonable but apparently controversial. Why use proven neurotoxins when safer alternatives exist? Why don't safety studies use truly inert placebos? Why give the same doses to 6-pound preemies and 20-pound full-term babies? Why don't we test for genetic susceptibility before injecting neurotoxins? Why did Congress remove legal liability if vaccines are so safe? Why do 54% of CDC employees end up working for pharmaceutical companies? These aren't anti-vaccine questions, they're basic quality control and safety questions that any responsible parent might ask about any product being injected into their baby. The path forward: what needs to change? Based on this investigation, several improvements seem both necessary and achievable. 1. Independent safety testing using truly inert placebos and long-term follow-up. 2. Mandate safer alternatives when they exist and are proven effective. 3. Individual risk assessment including genetic testing for detoxification capacity. 4. Weight-based dosing instead of one-size-fits-all. 5. Restore manufacturer liability to incentivize continuous safety improvements. 6. End regulatory capture by prohibiting revolving door employment. These changes would protect both children and the integrity of vaccination programs by ensuring they meet the highest possible safety standards. Conclusion: An informed choice. This investigation doesn't tell you whether to vaccinate your child. That decision belongs to you and your family. But it does reveal information that might not be readily available through conventional sources. The evidence suggests that current vaccines contain ingredients that wouldn't be acceptable in our food, water, or consumer products, yet they're injected directly into developing babies. Safer alternatives exist but aren't used. Safety studies have significant methodological flaws. Legal immunity removes incentives for improvement. Whether you choose to vaccinate or not, you deserve complete, honest information about what's being injected into your child. You deserve to know that safer alternatives exist. You deserve to understand why our current system prioritizes manufacturer profits over children's safety. Most importantly, you deserve the right to make a truly informed decision based on complete information rather than pharmaceutical marketing disguised as public health policy. Note on medical professionals: doctors are educated in medical schools funded by pharmaceutical companies. They learn what they're taught and repeat what they've been told. They're not independent researchers examining vaccine safety data, they're following protocols and guidelines created by the same industry that profits from vaccine sales. We're specifically talking about practicing MDs at hospitals and clinics who administer vaccines, not research scientists with PhDs who actually study vaccine safety data. Additionally, official medical research confirms that doctors receive financial incentives tied to vaccination rates. Studies show cash bonuses for practice-wide vaccination increases improved immunization coverage by 25.3 percentage points and that a bonus is a powerful incentive. Insurance companies use value-based contracts where doctors get extra payments for hitting 80% plus vaccination rates, with bonuses typically adding up to $100,000 or more per year per physician for meeting vaccination benchmarks. These aren't conspiracy theories, they're documented in peer-reviewed medical literature. Expecting hospital doctors to provide objective vaccine safety information while they receive substantial financial rewards for vaccination compliance is like expecting a McDonald's employee to give honest assessments of processed food ingredients. As parents, we research car seats, cribs, and baby food ingredients with meticulous care. Shouldn't we apply the same scrutiny to substances being injected directly into our babies' bloodstreams? The choice is yours. But now you have the information to make it with your eyes wide open. My personal decision, since many will ask what I decided after this investigation: I will not be vaccinating my baby during their first year of life. The risks are too high for me personally. I refuse to inject 4 to 5 milligrams of neurotoxic aluminum, multiple doses of carcinogenic formaldehyde, and blood-brain barrier penetrating compounds into my infant's developing system. I may consider selective vaccination later in childhood, after their immune and detoxification systems have had years to mature, but not during the vulnerable first year when these systems are least equipped to handle such toxic loads. This is my personal decision based on the evidence I've uncovered. Your decision is yours to make with the same information. The ultimate question: who is responsible for this bottleneck? After examining all this evidence, there's only one question left for every parent, every citizen, every individual to ask themselves: who, specifically which individuals, not faceless organizations, is responsible for maintaining this system that prioritizes profits over children's safety? We have documented that safer alternatives have existed for 25 years with FDA approval. These alternatives often work better than current toxic ingredients. The FDA can fast-track completely new technologies in months when motivated, yet the same agencies claim regulatory barriers prevent implementing safer ingredients. This isn't happening by accident. This isn't some mysterious force of nature. This is the result of specific decisions made by specific people who have names, faces, and financial interests. Public transparency and identified responsibility are essential for meaningful reform. The individuals, not corporations, who need to be held accountable include CDC officials who've spent decades moving between the agency and pharmaceutical companies. 54% of them end up working for pharma. FDA employees who explicitly tell departing staff they can work behind the scenes to influence decisions after joining. Industry advisory committee members who receive hundreds of thousands of dollars from vaccine manufacturers while supposedly providing independent guidance. Pharmaceutical executives who choose to use cheap toxic ingredients when safer alternatives exist. Regulatory capture architects who design the revolving door system between agencies and industry. Why are these individuals doing this? The answer is as simple as it is disturbing. 1. Personal financial gain through industry employment after government service. 2. Corporate profits from using cheapest possible ingredients without liability. 3. Career advancement by serving industry interests rather than public health. 4. Legal immunity that removes all consequences for their decisions. The system works exactly as designed. Babies get injected with neurotoxins and carcinogens. Manufacturers profit without liability. Regulators get rewarded with lucrative industry jobs. And parents are told to trust the science while being denied access to the actual science. This isn't a policy disagreement or scientific uncertainty, this is a deliberately maintained system of organized harm against the most vulnerable members of our society. The question every individual must ask themselves is, will we continue to allow specific, identifiable people to maintain this system, or will we demand accountability from the individuals responsible? Because at the end of the day, corporations don't make decisions, people do. And those people have names. Note, this investigation represents months of research into peer-reviewed scientific literature, official government documents, and regulatory filings. All sources are cited and can be independently verified. The author encourages readers to examine the primary sources and draw their own conclusions. Medical disclaimer: this investigation is for educational purposes only and does not constitute medical advice.